Hi, welcome everyone. Thanks for joining us today. I'm Alex Wills, Director of Fundraising at StartEngine, and I'm excited to host this conversation. Joining me today are Neil Thapar, CEO, Chief Science Officer, and Board Chairman of Barricade Therapeutics, and Scott Jordan, the company's fractional CFO. Barricade is a development stage oncology company based in Fort Worth, Texas. They're developing BT-1501, an oral, once daily, small molecule drug candidate designed to target the APC mutation, present in approximately 80% of colorectal cancer cases, for which there's currently no FDA-approved targeted treatment. Today, we'll cover the unmet need in colorectal cancer and why existing treatments are falling short, how BT-1501 works and what the preclinical data shows, where the program stands today, IND-enabling work complete, phase 1, anticipated second half 2026, the current Reg CF offering on StartEngine and how to participate, and for you to bring your questions. The team will take live Q&A during the session. Before we begin, 2 important disclosures. This Reg CF offering is made available through StartEngine Primary, LLC. This investment is speculative, illiquid, and involves a high degree of risk, including the possible loss of your entire investment. BT-1501 is an investigational drug candidate that has not yet been approved by the FDA and has not been proven safe or effective in humans. And here's the full disclaimer here. So I'm going to take it right to Neil and start this. Can you provide a little bit background on the unmet medical need in colorectal cancer?
Sure, Alex. So the unmet need in CRC continues to be a serious challenge as the rates of diagnoses and deaths are on the rise. And as this graphic shows, the statistics from 2023 with approximately 2 million cases, new cases of CRC, and approximately half that number resulting in deaths worldwide. And as you can see, the projections through 2040 are not improving. And with an estimated 1.6 million new cases and approximately 1.6 million people succumbing to this disease, Barricade's novel candidate for CRC, we hope to change this.
How does your drug work at killing these cancer cells?
So here I'll give you a very high-level overview of our drug, BT-1501. So BT-1501 binds to a protein in the body called epitope binding protein, or EBP. And specifically, our drug candidate will target cancer cells that harbor the APC gene mutation. And this leads to apoptosis or cancer cell death of those APC mutant cancer cells. Now, it's interesting to point out that while we also target healthy cells to some extent, those healthy cells are able to respond and compensate from that EBP inhibition. And lastly, with our class of EBP inhibitor drugs, normal cells are able to compensate from BT-1501 inhibition, whereas the APC mutant cancer cells cannot. And as shown in the last point 3, we've conducted the required safety studies by the FDA to show that a drug is potentially safe in humans.
So you've talked a lot about APC mutation. What is it and why does it matter for colorectal cancer patients?
So good question. So the APC gene it's a gene, right? And it stands for adenomatous polyposis coli. When this gene is in its normal state, it acts to maintain a healthy colon. And when the gene becomes mutated or truncated, it takes on a gain of function where it can drive tumor growth. And as you can see on the mid to right section of the slide, this leads to adenocarcinoma or cancer formation. Now, also you have loss of tumor suppression, as this indicates. And a couple of eye-opening statistics are, one, that 80% of CRC patients will present with this gene mutation, and 2, people born with this mutation have a 100% chance of developing CRC later in life. And those people are patients that have FAP or familial adenomatous polyposis coli. And one thing I like to address here on the bottom of the slide, when this gene is in its normal state, it maintains a healthy colon, right? It's the gatekeeper, if you will. When that gene becomes mutated, it loses its ability to fight off cancer. And as this time continuum shows from left to right, there's an onslaught of other mutations that can pile on, such as SMAD4, TP53, and PIK3CA mutations, and that leads to cancer and the spread of cancer or metastases.
Neil, what preclinical data do you have that shows your investigational drug works in colorectal cancer? And how does your drug compare to the standard of care chemotherapy used in these patients?
It's a good question. So as I just mentioned, people born with this mutation have a 100% chance of developing CRC in life. Well, the same is true in a genetic mouse model of CRC. So on the left section of the slide here, we have a genetic mouse model called CPC APC. And what we show is that the TASIN-treated animals did far better with very few polyps compared to the untreated animals. And here we treated these animals for only twice a week for 90 days. So very nice readout in terms of the activity of our drug. Now, in terms of how our drug works compared to standard of care, we've also studied our drug in the most heavily mutated model of colorectal cancer. And this is from a patient cell line known as DLD1. So this patient had the genotype with several gene mutations, as you can see here on the right side of that box, the APC gene mutation, KRAS, TP53, and PIK3CA. So those mutations in combo is the most recurrent genotype in stage 4 CRC patients. And as you can see, the orange trace or the orange line in the graph shows that the tumors responded. They were much smaller in size compared to untreated animals, as you can see with the blue trace, relative to standard of care on the lower right of the slide, showing that our drugs do as good or better from a tumor growth inhibition standpoint without the life-threatening side effects of chemotherapy.
Can you explain box 2 again? Just wanted to make sure the audience understands that again, please.
Sure. So box 2, this is where we have the DLD1 patient cell line. So DLD1 stands for Daniel L. Dexter, was a patient back in the 60s or 70s that came down with colorectal cancer, advanced CRC. And the graph shows the TASIN-treated animals, right, receiving our drug for about 18 days. The orange trace shows how the tumors responded to our TASIN drugs, showing that we had 63 to 74% tumor growth inhibition. Versus untreated animals, the tumors just didn't respond, obviously. So here we show a nice advantage to our drug. And compared to standard of care, which is in the lower right, the drugs that are used first and second line chemotherapy, like fluorouracil, 5-FU, in short, Erbitux, which is a anti-angiogenic drug, and also irinotecan, those are the 3 drugs, or of the 3 drugs that are used for first and second line standard of care chemotherapy for patients with CRC. And if you look at the tumor growth inhibition, or TGI, our drugs do as good or better. And to point out again and highlight that we don't have the same toxicities that those chemotherapy drugs do. Is that clear?
Got it. Yes, thank you. So what type of colorectal cancer patients will Barricade study in phase 1 clinical trial?
So in line with the xenograft data I just presented, here we had a patient, DLD1 for short, that had the same mutation in patients that present today in 2026 with CRC. So we will target stage 4 CRC patients here in red that have advanced metastatic disease, where even with the third line chemotherapy, which is Lonsurf and another chemotherapy drug in combination, they still have only an 8% chance of surviving 5 years. So we have to try something new. We have to do something different, change how we're treating these patients, at least in the third line. And again, the stage 4 patients in the call out box on the right shows that the most recurrent genotype is the one that we're targeting, the APC mutation, KRAS, TP53, and PIK3CA, which is the exact model we've studied historically with our TASIN drugs preclinically.
Got it. And tell us about the 17 million and CPRIT funding. What is CPRIT and what does receiving that level of support signal about Barricade's program?
Yeah, so this is where Barricade, we were very pleased to get these awards from CPRIT. So first off, CPRIT is the Cancer Prevention Research Institute of Texas, CPRIT, as you said. And they are a state of Texas fund, $3 to $5 billion in money that they can deploy to Texas-based companies that develop drugs in cancer, either prevention or specifically for oncology therapeutics. So we clearly fit those criteria. And just to give you a little history on our technology, so before Barricade acquired the TASIN portfolio from UT Southwestern Medical Center, the academics were awarded 2 academic secret awards. We then took it over and we started putting our TASIN programs through development, and we received a first $3 million seed award, which I'll touch on here, where we were able to execute the pivotal IND-enabling GLP tox studies with our drug. That allowed us to declare the clinical candidate as BT-1501. Those funds also enabled, in part, the manufacture of our clinical drug supply under GMP criteria. So now we have kilogram quantities of drug to get through the clinical trial. So that's a nice box checked. And then lastly, we had a pre-IND meeting with the FDA last summer, end of last summer, with positive feedback. So that sets a stage now where we can get that IND ready for filing. We'll talk about that in a little bit. But secondly, we applied for a second larger CPRIT grant of $14 million. And by the way, companies that apply for this grant, a very low percentage actually make it to the final rounds and actually get awarded. And that's between 10 and 15%, if I'm not mistaken. So we're very pleased with the fact that we're a double awardee now as Barricade. And the new grant will enable the development of our drug clinically in the phase 1 trial in the patients that I mentioned that have advanced APC-Mutant CRC.
And congratulations on that, Neil. I wanted to have a few follow-up questions to this just to help the audience understand this a little bit better. For one, can you explain just how and simplify how the non-dilutive grant requires a 2-1 match and how that works?
Sure. So Scott will talk about this in more detail, but what I can tell you is, so for the $14 million, we basically need to raise $7 million from non-secret sources, and that's from the public. And this is where our offering comes in here on StartEngine. Now, how we execute the grant, typically we don't have to raise all that $7 million at once. That would be preferred, but it doesn't need to happen in that way or in that manner. So for our first year, we need basically, call it $2.5 million in matching funds to deploy a certain amount of capital from CPRIT, which allows us to start the clinical trial.
Got it. Okay. Well, I'll have Scott go through that in more detail after this. I didn't mean to take his question away, but I just wanted to make that clear. It's good to repeat it too. And since we're running good on time, I wanted to see if you can help explain too a few of these acronyms too, because I think that will help better help retail investors understand this better too. Can you explain what IND and pre-IND means when you put that in here?
Absolutely. And I think we have a slide kind of that breaks that down. So maybe it's the next one, Alex. Take a look.
Okay.
Just to give people the visual. Yeah, perfect. So
perfect.
The first like here, what the IND is, it's the investigational new drug application. And this is what the FDA requires of pharmaceutical companies or early companies like us before we study drugs in humans. It's a very voluminous data package, right? Where you prepare everything that you've done with your compound. You've demonstrated that you can manufacture it under CMC, which is chemistry, manufacturing, controls. So John Walling is our SVP of CMC. He's done a spectacular job with that. He basically got us to the point of having kilogram quantities of a tablet at 2 different tablet strengths, at 25 and 100 milligram. Mm-hmm. Pharmacology, pharmacokinetics, toxicology, clinical aspects all feed into the IND application. So this IND submission is planned for July. We were targeting this month, but we just actually brought on a CRO to conduct. One of the important aspects of the phase 1 trial is establishing basically what the human's body does to the drug. In other words, the pharmacokinetics, and you have to have those data as in part with the phase 1 trial. So long story short, that's underway, and those validations will start later this month. That's the bioimmethod validation component under clinical. And just kind of to add on to this, Alex, at IND, the clinical aspects is a pretty huge component of the IND itself, right? You need to show the
Mhm.
FDA that you know what you're going to do in terms of the phase 1 trial, what your inclusion criteria are, your inclusion exclusion, what your patient population is. We've defined all of that, and that's very robust, and we're nearing the finalization of our clinical protocol itself. So yeah, in short, that's
Very,
basically the clinical aspect. Go ahead.
very helpful. Yeah, thank you. And I'm asking this question again. I'm just repeating it, but I think it's worth repeating. Just so, where would you say right now Barricade is on the path to clinical trials? I know this is repeating what you're saying. Just yeah.
Right. So right now we've got some heavy lifting on our shoulders and getting an IND buttoned up and submitted. That's what's going on behind the scenes. And once we get that IND filed, next month, we'll be in the position to get our clinical operations group fully engaged, get the site initiation visits and the pre-trial setup tasks accomplished with the various trial sites here in Texas, for example. And once the trial is underway, and I hope I'm not jumping ahead of you here, but we do plan to start the phase 1 trial in Q4, right? And that is, of course, contingent on us raising the necessary capital to unlock the $14 million secret award, which Scott will dive into. The second bullet here, in terms of the readout, right? We envision an active dose, basically 10 months from the first patient in, or basically from the dosing of the first patient in the trial. So from a timing perspective, that's around 3Q to 4Q of 2027. And in order for us to get to the second largest phase of the phase 1 trial, which is the expansion or the 1B or the part 2 of the trial, we basically anticipate having data from that phase 2 years out, which is basically 3Q to 4Q of 2028.
Got it. Thank you for that. And yeah, I'll pass this question to Scott. Can you tell us a little bit more about the capital raise and StartEngine, please?
I think Scott's on mute.
Sorry. The company is raising a $1.235 million award from StartEngine. This is a Reg CF offering associated with raising capital from retail and accredited investors. The company will then expand the round, the Reg CF, to raise up to $5 million after completing a PCOB audit. And so we'll be utilizing the capital from the Reg CF to unlock the secret award, which we just discussed, and initiate the company's phase 1 trial.
Okay, thank you. And I know Neil already explained this as well, but can you explain how secret grants work and how does the release of the money work? What is the the 2-1 match? Again, I know we're repeating a lot of information here, but I think it needs to be repeated.
Well,
I
totally agree with the emphasis on the 2-for-1 match. I mean, what we're talking about here is we're talking about efficiency of capital, meaning that there's $14 million of non-dilutive capital, which can really impact investor returns if the drug were to be approved, because it's non-dilutive, which is a key aspect. So very few companies, biotech companies, have this level of non-dilutive capital. And the way that it works is that the company raises the match financing, utilizes that capital to purchase services or to accomplish certain objectives, and then quarterly the company submits to CPRIT for the monies under the award. So that's kind of the cadence. So with the 1.235 million, it unlocks 2.4 million in CPRIT grants. So instead of 1.235, the company has 3.7 million. This capital will be utilized to file an IND to the FDA, which we just talked about. It will also the full engagement of a clinical contract research organization, a CRO, which are utilized in industry for the clinical trials, perform site initiation at some very prestigious Texas-based institutions, and initiate the phase 1 trial, which is very important for determined safety primarily, hopefully with early hints or signs of efficacy, pharmacokinetics, tolerability, which is basically like, say, blood levels of drug and concentration in a tumor, And then obviously we want to get to patients with advanced CRC. Now the use of proceeds for $5 million, which is the total Reg CF round, unlocks $10 million in CEPA grant funding. That $15 million total will be utilized to execute the part 1 of the phase 1 trial and allow determination of the recommended dose for part 2 of the phase 1 trial. So the milestones, basically, this capital will be utilized with the Reg CF, just in summary, is to get the company through an IND submission to a phase 1 clinical trial. And if most investors, though preclinical, you cannot underestimate the importance of having really strong data like Barricade in preclinical. It's very exciting to know that the company is a matter of months away from an IND filing and entering phase 1 clinical trials in patients with advanced CRC.
And now, assuming that you meet this requirement in the next month or two, you will be updating investors and audiences on StartEngine about that, and through campaign updates and emails.
That's exactly right. So every time we hit a major milestone, we would highly encourage everybody that's on the phone right now to become a follower, to log into StartEngine, and to go to our campaign page and follow our page, because by doing so, you will receive campaign updates throughout. Right.
That's great. Yeah, and I think this is a great use case for that. So how do investors participate right now? Just wanted to go through that.
Yeah. So what it entails, there's 7 steps, but it's very, very efficient. It's a very nice system. This is the future of This is the future of those who are going to invest in private placements, like we've seen in the public markets with E-Trade and Fidelity and Schwab. We really believe that raising private placements online has significant advantages, and a few of the advantages is basically you can make an investment using your credit card. You can make an investment utilizing funds in your IRA, right? You can do due diligence online so that you understand the risk and the opportunities. And so everything culminates in submitting your investment, receiving confirmation, and monitoring that investment. And in the future, we may be looking at things like secondary trading, tokenization potentially, right? So very, very easy and transparent onboarding to make your investment. And our minimums are low, so that those who are more retail, non-accredited, can participate in potentially treatment for advanced colorectal cancer, which has incredible clinically unmet need, and those who are accredited investors that would like to invest more money.
That's great. Yeah, and this is the steps to do this. It's going to be on the campaign page at startengine.com. You can see it on the crowdfunded page. So I wanted to bring it back to Neil. We have some time. Let's get a few more questions in. And again, this is a topic that's hard for retail investors to understand. And there's definitely a communication gap between your understanding and how we're presenting this to retail investors, and understanding not only how the process works, but as Scott went through the funding structure as well. All that's really important to understand. For Neil, can you shed light on the pipeline you have right now in terms of tackling other types of cancers and other diseases?
Of course. So, as we've already mentioned and discussed, our lead program, BT-1501 and APC-Mutant CRC, right? The timeline we've already discussed, and that's shown here. Another pretty big opportunity for us, which is in line with BT-1501, and I did touch on this earlier, is to address the unmet need in patients that have FAP, or the familial adenomatous polyposis coli, right? So these are kids, basically. They're born with a mutation. They have polyps, tumors throughout their colon. And by their early teen years, these poor kids are dealing with resections or sections of their colons being removed and having to deal with a colostomy bag, right? That's not pleasant, and it's just a horrible disease. And based on the data that we have today, we believe we have a good shot at helping these patients. Now, the criteria here are this. We have to show that BT-1501 is safe and tolerable as part of our phase 1 trial. Now, once we hopefully show that to the FDA, our plans are to have another Type B meeting with the agency, with the FDA, and potentially open another IND for BT-1501 in patients with FAP. So in addition to the prevention measure here with FAP patients and the third line down, we also have the potential to explore other cancers, neuroblastoma, and affecting, again, the younger patients, which is a very brutal disease, orphan indication, Burkitt's lymphoma, and hepatocellular carcinoma. So there are publications out there with our early drugs, TASIN-1, as an example, that show our drugs could be effective in these cancers. And these are areas we wish to explore and help building out the pipeline. And then lastly, outside of cancer entirely is neurology. So Big Pharma is actually pursuing EBP inhibitors as potential druggable, targets in terms of drugging EBP to promote remyelination of nerve cells. And I'm talking about specifically the patients that have multiple sclerosis or MS. So that's a disease where if you take the nerves in those patients, they lose the protective coating or the myelin sheath around the nerves. And that's why they lose that muscle conductive capability, and they can't move, they can't walk, and eventually talk. It's a horrible disease. So what our drugs have been shown to do is rebuild that insulation. Take a wire, for instance. You have a wire and you have the insulating sheath around the wire. We're basically putting a new protective coating on that wire to allow the nerve conduction from the brain to the fingers, from brain to the hand. So again, this is another area we plan to explore. Just take some capital, and I think we'll have a very nice, robust pipeline for Barricade where we can build out the company in those specific areas.
And these are just a follow-up question from a layman. Would you say that in terms of these future potential treatments, would these also be potentially by a once daily pill?
That's a good question. There's a good chance they could be. Based on the data we have, we've dosed animals orally primarily, and that's for compliance and convenience. For switching over to cancer again, when patients go to the infusion clinic, those are lengthy processes and they're exhausting, they're toxic. And to have a pill that you could take orally once daily or even less frequently, possibly, that's nice. I mean, that improves quality of life, especially if you have a safer alternative to toxic drugs. But yeah, the goal and intention here is to either have a tablet or a capsule.
Maybe it's a little bit too specific, my question, to ask if it should be once daily in a pill, but you get the point. Yeah,
I do.
Yeah. And also follow-up too is that every use case that you see, a potential use case, like you mentioned with oncology, neurology, all of those would require those, from my understanding, separate IMD submissions, correct?
That is correct.
Got it. This is all good to know. A follow-up question. So why does the APC mutation in CRC, colorectal cancer, represent an attractive opportunity?
Yeah, so that's a great question. And this is where, I had this question a lot, over the years in diligence with various parties. And now we're starting to see that there is a huge potential here. So in CRC, for example, there's a lot of companies that are targeting other mutations that I mentioned earlier, such as the KRAS mutation highlighted the first row in green. Then you have BRAF mutations in purple and so forth, HER2 and MSI high. So those are all various mutations that occur in CRC. And you can see the percentages, 3 to 5% for KRAS, BRAF 8 to 10%, HER2 positive CRC 2 to 4, and then MSI high or microsatellite instability high, which has DNA mismatch repair, and that's very deep science-wise. But just the takeaway here is that is 5 to 15% of CRC patients. Okay, that's 5 to 50% of colorectal cancer patients. Bottom line, we are targeting 80%. So that's already the bulk of the CRC patients that we're focused on. And also this slide captures approved therapies. So here we have, it's an attractive opportunity where we're targeting the bulk of the CRC market, if you will, that has no approved therapies. We have a good shot goal based on the data that I've presented here. with a major unmet need. So these patients are underserved. They have been underserved for decades. Hope that
Is there
answers
an
your question.
overlap? Is there an overlap at all in these mutations, or is it just in to parcel out?
There's not really an overlap, but there are some tumor percent of variety, a mix of mutations. For example, APC and KRAS could occur together, and BRAF, we've shown that. So there is a potential down the road for strategies, which we definitely want to employ and consider. We're not trying to say that our drug is going to be the Ferrari, if you will, that it's going to, make everyone perfect and take away all the cancer. The reality is we're going to knock it down, and maybe in combination with some of these other therapeutics, we could see a greater or more efficacious response to the treatments. Does that make sense?
Yeah, it makes sense. And what you're mentioning before about this being 80%, this doesn't include the unfortunate growth in CRC incidence among patients. I know you're not really touching on that because that's not really in your control, but that's probably something worth mentioning, right?
And that's a valid point and worth mentioning too, because over the last 5-6 years, we're seeing a drastic rise in early onset CRC, right? Early onset, what does that mean? We're talking people in their 40s and 50s having colorectal cancer, and not just stage 1 or 2, but more advanced colorectal cancer. So that's an eye opener for me, I can tell you that. So it's nice to have that potential to hopefully have a therapy that's safe, effective, and get it into patients as fast as we can.
It makes sense, yeah. And are other pharmaceutical companies developing EBP inhibitor drugs like yours?
Right. So, and that's another big question we've had over the years is, is big pharma developing drugs like this? And so now we can say comfortably, and it's nice to say this, yes. So Sumitomo Pharma is a good example of this. They're a Japanese company. They now have 2 clinical trials. One has been open for a couple of years now. On the left, it's their drug DSP-0390. It is an EBP inhibitor given orally, but they are targeting patients that have a form of brain cancer, and they're calling it here high-grade glioma. And just to skip right down to the punchline here, in this study, they've already demonstrated activity where 2 patients that have a specific gene mutation, and again, we're not targeting brain cancer, but I can tell you that the patients that responded had IDH mutations with grade 3 gliomas, and they had tumor reduction. So that's a pretty big deal. So typically when patients are diagnosed with brain cancer, it's not a good sentence, if you know what I'm saying. We're talking 6 to 12 months of life at best. And to see that these patients have been on drug for more than 9 months, that's promising. Now, if I could kind of switch over to the right, their other trial. So they observed some modicum of activity in that first trial. And now Sumitomo is opening up another, and not brain cancer, but small cell lung cancer patients. And there's a brief description. We'll have this deck shareable, right, for the audience to look at later. So I'm not going to read it word for word, but they're planning to start this study in July of this year. So the takeaways are for Barricade, we're not directly competing with Sumitomo, but rather Sumitomo now has basically validated EBP as a druggable target in cancer. And then in addition to that, they've also shown that their EBP inhibitor is safe. It demonstrated pharmacokinetics, the PK and PD components of pharmacokinetics and pharmacodynamics, and that the drug was well tolerated in patients. And those are huge selling points when you're trying to target cancer, right? Because as we all know, chemotherapy does not have a good rap. The image of patients on chemotherapy is bad. And then lastly, the point I want to make here too is that, while, we are targeting a form of cancer, not brain cancer, we feel that Barricade is uniquely positioned to become the first precision therapy for APC-Mutant CRC. And, our IP supports and backs our indication going after colorectal cancer.
We had a question from the audience. Would you say, why has APC mutation been ignored for so long?
That's a great question. I think they have tried to drug it in the past and not had success, but now there clearly is an indication that it could be successful. Why they wait, I don't know. We'll find out maybe now. There's going to be, once we get to the clinic, I think we might create a buzz and an awakening, if you will, in pharma that will push that to the, with pharma companies jumping in.
And you view it as Sumitomo, what they're doing as not only validation, but it's a benefit for Barricade Therapeutics outside of the obvious benefits for patients, but also for your company.
I do. And just for the mere point, those points highlighted here in blue with the checks, one of the big things is the risk for drug development is when you go into your first in human clinical trial, the question is your drug safe, right? Will patients tolerate it? Are you going to be able to demonstrate PK, PD? And just to be clear on what that is exactly, the pharmacokinetics is, again, what the body does to the drug. So you have levels of your drug in blood over time. And then the next question, two-part question for PK, PD is, what does your drug do to the body, right? So it's going to hit a receptor. We're going to hit that protein. We're going to knock down that protein, EBP, and it's going to trigger a response. So there's your PK-PD relationship, and Sumitomo has demonstrated that. So it's nice for us, and we're going after the same biomarker from a target engagement perspective, which is really nice because we've demonstrated that in our preclinical models. I really can't speak to it because it's probably confidential for Sumitomo, so I have to be careful here. But from a Barricade perspective, we're pursuing the exact same target engagement biomarker, as it's called, for PD determination.
Okay. And I would say, I know we're done with all questions, but I think it's also just worth going through your background too. We didn't really get to go through that. Neil, would you be able to kind of give your background and what you've been doing? And
Absolutely. So I'm a clinical pharmacologist by training, and I received my undergrad from the University of Western Ontario in Canada, London, Ontario, Canada, in biology and pharmacology. I then went on to get my graduate degree in pharmacy at University of Houston. So I got my doctorate at U of H, and then went on to do a postdoctoral fellowship in clinical pharmacology at University of Texas, MD Anderson Cancer Center. That was a nice stint, a 2-year stint at MD Anderson. And then I worked at Anderson on staff in their clinical pharmacology department, and I rounded with the clinical team, did a lot of R&D in the lab. So it was a very nice learning experience at Anderson. And then after that, I was brought on to Reata Pharmaceuticals, and I headed up the early development of their preclinical cancer and type 2 diabetic nephropathy programs at Reata. And ultimately, Reata was acquired by Biogen for about 7.3 or 4 billion just a couple of years ago. So that was a nice little exit for everyone involved. And since that time, I've always had the motivation to start a company around a solid technology, work with a solid team of scientists here at UT Southwestern, and co-founded Barricade in 2018, 2019, and got this program up and running. And just so everyone knows, BT-1501 did not fall in our hands. We had to basically work and generate that. And, our chemist, John Walling, did a spectacular job in arriving at this analog, and we all worked together and understood the pharmacokinetics, the safety, the tolerability, the toxicity of the molecule. So yeah, it was a fun. Hopefully that's a good enough background on myself.
Yeah, it is. Thank you. And yeah, I just wanted to I know you have some board members too that are worth mentioning as well. I know I'd love to go through the whole team, but for time's sake, is it's worth mentioning any board members too as well?
Certainly. So we have Darlene Boudreau and Carlos Guillem. So Darlene and Carlos are both involved in drug development. Darlene serves on the boards of other early stage biopharma companies. She had a nice little exit with Mucinex. I'm sure everyone has taken Mucinex or guaifenesin.
I've seen the commercials.
Yeah. So she was involved with that. So there's a lot of history here. And then Carlos has done a lot for other Texas-based biotech companies. He's on the boards of other companies as well. And so to have them involved with Barricade from a strategic perspective and guidance, it's huge. It's very impactful for us.
That's great. Thank you. I don't want to exclude any of your team members. I just thought it was worth mentioning your board because you have a strong board. And Scott, just real quick, just real briefly, how have you been involved in biotech companies in the past, in your background?
Yeah, so 30-year track record, started my career at Abbott to C-level executive. I'm currently the chief financial officer at Barricade Therapeutics, also investment banking to a company public via NASDAQ. And great to be here. I want to thank all of the those who decided to attend our webinar. Really appreciate it.
Yeah, Neil Scott, really appreciate it. Thanks so much for your time today. It's really been really informative. And yeah, we'll see if we can get this shared out as a campaign update, I would say, on your page. Just update the audience on that, please. And yeah, thank you for everyone joining. And as a reminder, this Reg CF offering is made available through StartEngine Primary, LLC, member of FINRA and SIPC. This investment is speculative, illiquid, involves a high degree of risk, including the possible loss of your entire investment. BT-1501 is an investigational drug candidate that has not been approved by the FDA and has not been proven safe or effective in humans. Thank you. And, if anything else comes up, let me know. But really appreciate being here. Thank you.
Thank you all.
Thank you so much.